So the ABO system, we now know about universal recipient, universal donor. You must know that the antibodies which are made in the ABO system belong to the IgM class, immunoglobulin M class. Okay, so what is unique about the IgM antibodies is this is very important. It's a very large molecule. It's actually got 5 antibodies stuck end to end like this. So you've got 1, 2, 3, 4, 5 antibodies stuck end to end like this. And this has a lot of, you could say, valency, ability to bind to different RBCs. Of course, RBCs are actually really big, and you won't exactly bind to 10 over here, because if this is one antibody, they actually bind to 10 antibodies through a joining fragment that links them all together. The point is, IgM antibodies help in coagulation and clumping of blood. It causes good clumping, or agglutination, L-U-T-I-N-A-T-I-O-N, agglutination of the blood to take place over here. So that is very important, IgM. Why? Because IgM does not cross the placenta. Because if you see a woman who is, let's say, A blood group, she marries a person with B blood group, and they have an AB baby, the antigen of the baby is different from that of the mother. There could be a chance of reaction. That doesn't happen so easily because the mother's antibodies, which she makes against the baby, cannot cross the placental barrier, because it doesn't. Okay. Then after that, you must know that there are other, the other major blood group system is the Rhesus system. Rhesus, Rhesus. This was found in the Rhesus monkeys, so it's very important to know about also, because we have this antigen. If we look at the world, the Caucasians are around 85% Rh positive. They have the D antigen, which makes you Rhesus positive. Actually, there are 3 antigens, C, D, and E. You can be positive for these antigens, and that makes you Rhesus positive. So you might be negative, that's small c, or you might be positive, capital C. Again, this depends upon your genetics, small d or capital D, small e or capital E. All of these, you have different permutations and combinations. You can have D, whatever, mix and match over here, mix and match. The point is, we mostly focus on the D antigen, and when one of your chromosomes has the capital D, the D antigen on it, you are Rh positive. 85% of Caucasians are this one, of whites, of Caucasians, C-A-S-I-A-N-S. Caucasians are the whites, the the European race. They are 85% Rhesus positive, whereas the Asian people, the Asians over here, are around 90, more than 95%, 99% positive for Asians. Like that, like as Indians, Chinese, Middle East, to an extent, all of them have this. They're more commonly Rhesus positive. Okay, and the most important antigen is the D antigen. Okay, now in Rhesus, if you are Rhesus positive, you have the antigen called the D antigen on your RBC. So that's another system we're talking about over here. Now then, generally you will not have the antibody. That's Landsteiner's first law. It's followed over here. If you are, however, Rh negative, okay, so you are not D positive, you are D negative, Rh negative. So in Rh negative blood groups over here, these people will not necessarily have antibodies against the D antigen. They don't always have that. Remember this. So what happens is, when this person who is Rh negative gets exposed to Rh positive blood sometime in their life, that is when they will make those antibodies against it. Please remember this. So if, for example, a mother is Rh negative, and by the way, the antibodies here belong to the IgG class. IgG antibodies can cross the placenta. Very important. They can cross placenta. the placental barrier. This is the only antibody that can cross the placental barrier, playing a very important role in passive immunity, where the mother can transfer her antibodies to the baby, and it gives the baby some safety for up to 6 to 9 months. By that time, he gets vaccinated for most of the big infections that take place and affect the child's health. Anyway, so the point is, now if a mother is Rh negative and she marries an Rh positive person, so a mother is Rh negative and she marries, so the father is Rh positive, for example. So if they have a baby who is having one of these D antigens, then these D alleles, you have got an Rh positive baby. Now it's in the mother's body, so the mother is pregnant with an Rh positive baby. Generally, what happens is during the first pregnancy, it's not a big deal until she delivers the baby. In most of the cases, 96% of the cases, when the mother delivers the baby, like the baby's almost already out. That time, there's a little bleeding that takes place in the placental barrier. And that bleeding is where the blood of the baby and the mother mix. And that's when the mother, who is Rh negative, gets exposed to Rh positive antigens for the first time in her life. And that is what's going to go on, and she will develop antibodies against it at that time. And so now if she gets pregnant again, these antibodies, these anti-D antibodies, are floating in her blood, and they can go to the second or third child and affect the child. So the second child has a almost 50% chance of getting affected, and the third child has an almost 95% chance of getting affected, like this. So the more pregnancy she has, the more sensitized she is to the Rh antigen, and she makes these antibodies accordingly. And the worse the prognosis for the next child and the next child. And I'll talk about this in more detail. It's called the hemolytic diseases of newborn, where there is Rh incompatibility. We call it Rh incompatibility between the mother and child, and we will discuss that in a little while. So remember, Rh blood, IgG, crosses the placenta. Okay, you don't always it doesn't follow the second postulate. Other minor blood group systems that exist, other minor blood group systems are, for example, you have these systems like the P system, the MN system, the Kell system, the Duffy system. Then you have some familial systems like Diego and Lewis. Now, these systems are not, okay, we can also type in Lutheran, Kid. I know. So these are like family names. Most of these are family names. So they were discovered in women while they were going through childbirth at that time. So they're named after their family name over there. Okay, let's just talk about a few of these features. See, these antigens are not just there to prevent other blood from mixing over there. It's also important in what do you say? They have some clinical aspects you should know about. Like, for example, if you look at the Duffy system, if you are Duffy antigen positive, this antigen is actually a receptor for malaria parasite, for your plasmodium vivax, VIVAX. This is a protozoan that causes malaria. Okay, and so when it enters the body, it tends to stick to these receptors and find its way inside, or the cell much easily, inside the red cell much easily. Duffy antigen receptor for cytokines is absent in these people in some people, and that leads to mild neutropenia. So they can have a mild neutropenia if you are absent, if you are deficient in this antigen. Okay, then after that you have another system called the Kell system. Kell system. Now, if we look at the Kell system, it's a very important system. Like after ABO and RH, even though these are minor systems, meaning they don't tend to react, this has a little higher probability of reacting. Okay, next to ABO and RH blood group system. So it is the third most important blood group system where you. What makes something major is if you have a mismatch, there's a major chance of blood transfusion reactions taking place. But in minor, not that much. However, if you had to say which is the most likely to give you a reaction, Kell comes over there. Kell. So what happens is these people, if you don't have the Kell system over there, absence of the Kell system leads to something called MacLeod phenotype. MacLeod phenotype. Meaning to say that they have got this one, RBCs with spines. So their RBC shape is got these little spines on them. It's not that smooth round surface over there. RBCs with spines, we call them acanthocytes. And then you also have a reduced RBC survival rate. So your RBCs die faster. They don't survive exactly for 120 days. And you get this muscular dystrophy and cardiac defects associated with that. So the muscles tend to get weak, and the heart has many defects over there. In the P system, these are where you have autoantibodies. They are called Donath-Landsteiner antibodies. Donath-Landsteiner antibodies. And they what happens is when a person is exposed to cold, they get this thing called paroxysmal cold hemoglobinuria. Paroxysmal cold hemoglobinuria. So they start peeing hemoglobin in their urine. Why is that? Because when they're exposed to cold, it causes an immunological response, and there will be destruction of the red cells, and the hemoglobin is released, and it gets excreted in the urine. Okay, these types of antibodies are seen especially when these people are infected with syphilis. The P antigen is a source of where there is it acts like a receptor for a certain virus called parvovirus B19 and E. coli, and for the E. coli. So what is parvovirus B19? This is a virus that suppresses your bone marrow. Okay, if a person has some hereditary anemia like thalassemia or sickle cell anemia, and they get affected with parvovirus B19, their bone marrow is not going to work. It's not going to produce new red cells, and there will be a sudden massive drop in their hemoglobin, and that could be life-threatening, especially in people with sickle cell anemia and thalassemias over there. Okay, so the MNS system. The MN system is used for you can use it for those paternity tests, paternity disputes regarding who is the parent. Is this child really that person's child? So before we had the DNA testing, genetic testing, we used to use blood group systems to get a clue as to whether this child is really this child in case of hospital mix-ups, or in the case of very rocky marriages, you could say. Now, how can we use blood groups in such cases, like to decide if the child is this person's child or not? Now, if a person, for example, is AB, I'm just giving an example. Her father is AB and the mother is O blood group. So she is O blood group. So you got two chromosomes which are O blood group, one which is A, one which is B. Now, you can get a B blood group baby, you can get an A blood group baby, but you cannot get an AB blood group baby. If the baby came out AB, that means something is wrong. Maybe they mixed up the baby in the hospital. You understand? Because you can't have AB blood group if one of the parents is O blood group. Okay, like you are doing this as a diagnosis of exclusion. Okay, now, for example, if a person was having, for example, an affair, just as an example, so here's the mother is AB, and the father is O blood group, for example, O blood group. Now you can get A and B blood group. Okay, you can get A, you can get B, babies over here. But however, if the mother had an affair, for example, with another man who is also O blood group, you can end up with the same permutation and combination. So we cannot say definitively if the child is your child, but we can definitely say if a child is not someone's child, if the blood group systems do not match their Mendelian genetics over there. Okay, now some other interesting points I just wanted to go over. Ria's blood group, if we look at it, these IgG antibodies that you see over here, they don't usually what do you say, cause too much of clumping. They don't clump too much, or they don't cause agglutination. So they're called incomplete antibodies. So these IgG antibodies are incomplete antibodies. What happens is they will stick to the cell, but if you want to see clumping take place, you have to either add some special antibodies against these antibodies. So here's a cell, it's Rh positive, so this is the Rh antigen. Okay, so humor me, this is the Rh antigen. When you mix the antibodies with the Rh antibodies, they're naturally going to stick, because that's what, it's named after that. So it gets stuck. So you get the Rh antibody, but there's no clumping. It's not going to clump that much. It's not having that valency like the IgM antibodies, because these are IgG antibodies. However, if you stick anti-immunoglobulin antibodies to this, so they stick to the immunoglobulin over here. So now they can bring more of these antibodies together, and they cause more clumping that time. So when these are called Coombs antibodies, we call them Coombs antibodies over there. Okay, and otherwise you can also do one more thing, which is you can give albumin. You can mix albumin to that mixture. So by mixing albumin, it potentiates these antibodies to function better, and that causes clumping again to happen effectively that time. So it's just something to remember over here. So when we do blood testing, we for your blood type, we actually try to use the IgM class of antibodies, which are made in the lab for these testing purposes over here.